Hello, this is hopefully what is a relatively short video on human blood groups. Before we get into the blood groups themselves, you need to understand this term right here, antigen. You need to know what an antigen is. So your immune system produces antibodies. They are proteins that stick to stuff that's not supposed to be there and label it as foreign. The thing that an antibody sticks to is called an antigen. So the definition antigen is a functional one. It doesn't matter whether it's a carbohydrate, a protein, or some mixture of the two like cartilage. Anything an antibody sticks to is called an antigen, and this is going to become important later. So then specifically when talking about blood, we have these different ways of grouping blood, and there are all sorts of different ways to group the blood. The most important ones are the ABO blood grouping and the RH blood grouping, RH positive or RH negative. We're going to do each of them separate and then bring it all together. So you've got your ABO blood groups, which is based on the presence or absence of a number of different glycoproteins called agglutinogens. So these are just proteins that are on the surface of your red blood cells. You are going to see later that these proteins have the potential to be immunogenic, or they can be antigens and stimulate the immune system or be recognized by the immune system of other people. So you might have on a red blood cell, let's just draw a red blood cell, right, all sorts of proteins, but most of the proteins on my red blood cells are exactly the same as the proteins on other people's red blood cells. So nobody's immune system responds to them. But the ones that come in different flavors, that your immune system recognizes, that we call agglutinogens, those are important because they interact, or they have interactions with the immune system. Okay, so how does it work? There are two different agglutinogens, the A agglutinogen and the B agglutinogen, but as you can see here in this line, there are four different possible blood types. Or ABO blood groups. This is because you inherit two genes for your agglutinogen. You get an agglutinogen gene from mom and an agglutinogen gene from dad. If genes come in different versions, we call them alleles. Right? So every human being on the planet has all of the same genes, but we get different versions of those genes called alleles. So there's one piece of DNA out there that codes for the B agglutinogen. There's a piece of DNA in the world that codes for the A agglutinogen. And there's a piece of DNA out there in the world that doesn't work. So you are going to inherit two of these three different alleles. Right? So if you get, let's say, the A, you can get two copies of A, or A and O. That makes you type A, because this gene, or this chromosome, when you inherit this piece of DNA, this allele, this version of this gene, makes the A version of the protein. This version here makes no protein. So there is no O agglutinogen. O is the absence of agglutinogen. So again, if you're type B, this means you have the B agglutinogen gene and either two versions of B, or your B and O, and the O chromosome or your O gene just doesn't do anything. So let's say you inherit B from dad and A from mom. Now you're AB. Both of those genes are going to be expressed, and you'll have both proteins on the surface of your cell. If you get an O protein from mom and an an O gene from mom and an O gene from dad, then you have two non-functional genes or non-functional alleles that you have inherited, and you are going to make neither of the agglutinogens. So the way your immune system works is that, sorry, I needed to adjust my headset. Your immune system produces antibodies to anything that is foreign. In actuality, what your immune system does, and it's a pretty amazing numbers game, is that it just randomly makes as many different shapes antibody as it can dream up, because your body doesn't know what's out there. Right? If you are type A, your body is not aware in any sense that there is a B protein out there that isn't supposed to be in the body. It just makes antibodies of all different shapes in the hopes that when something foreign shows up in your bloodstream, one of your randomly manufactured antibodies is going to be able to stick to it. And if your immune system has developed properly, it'll only stick to things that aren't supposed to be in the body. So theoretically, you have antibodies to any antigens that don't exist in your body, which means you produce antibodies to all of the agglutinins that you may not have. These antibodies, all the agglutinogens that you may not have. These antibodies are called agglutinins. So the cell surface protein is called an agglutinogen. You produce antibodies called agglutinins, right? And it is the agglutinins that are going to recognize the agglutinogens. So the agglutinogens in this scenario here are antigens. They're being recognized by the antibody. And then, so the way it works is, let's see, I've got red. If you're type, oh, we already said, right? If you're type A, this means you have the A protein on the surface of your cell. And if you're type B, you have the B protein. If you're AB, you've got both of them. There's an upside down A, right? And if you're type O, neither the A nor the B are there. So you can put it all together in this figure here. If you're type A, you make the A antigen, and your immune system will make antibodies here that recognize the B antigen, because it's not supposed to be in your bloodstream. If you are type B blood, then you have the B antigen on the surface of your red blood cells, and then you are going to produce the anti-A agglutinin. So B agglutinogen, anti-A agglutinin. If you are AB, you are going to have both of the agglutinogens here, but neither of the agglutinins, because your immune system is going to be programmed to not recognize those two potential antigens. And then if you're type O, you have neither the A nor the B, so your body is going to have the ability to produce antibodies that recognize both A and B. Don't worry about this part down here. We are not doing who can donate to who, but it's fun to think about. So we can look at it real quick. But basically, you don't want to get any blood that has an agglutinogen that you don't have. So because O has no agglutinogens, anybody can receive from O, right? Because there's nothing on the surface of O for anybody's immune system to respond to. So that's why everybody has O here. So if you're type A, you can get A blood and you can get O blood. You don't want B or AB blood because it's got the B agglutinogen on it that your antibodies are going to react with. Right? If you're type B, that means your bloodstream is anti-A. It can't have the A protein in it. So that means for type B people, B and O can donate, but no A and no AB. Then if you're AB, lucky you, right? Your immune system has learned or been programmed to ignore both the A and the B agglutinogen, so you can get blood from anybody, A, B, or O. Then, so this is important because if you put the wrong blood in the wrong person, you can get a transfusion reaction, or what is called agglutination, when the antibody agglutinins stick to the cell-bound agglutinogens and cause clumping or cross-linking. So you want to think of, right, you'll have a cell here that's got an antibody attached to it, which is attached to another cell, and these antibodies are actually really big. I'm not drawing them accurately, but it doesn't matter. And you can just imagine antibody, 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 blood cell, blood cell, blood cell, blood cell. So you get these big clumps of blood cells all cross-linked together with antibodies. On a slide when you're doing the test, it looks like this. You can imagine if your blood got all clumpy like that in your bloodstream, that might cause some problems. So this is why blood grouping is important. There is an additional important way to group the blood, and that is the RH blood groups. So this is just another agglutinogen, another surface protein, which is potentially immunogenic. So it can act as an antigen if it gets into the wrong person. So there's a whole family of RH agglutinogens. I don't know how many genes are involved, so I don't really understand the genetics of this. If I'm understanding it correctly, it is just one gene that has 45 different alleles. It would be like if there were 45 different eye colors or something like that. But at any rate, those are Rh factors. So it's just another protein that we need to track, and we want to know whether it's there or not there. The most common Rh factor is the DRH factor. So a lot of times, like if we look back here, it says anti-D. They're probing for the D version of that Rh. So then, if somebody is Rh positive, it means they have the Rh protein, and you give them a little positive next to their blood type. If they're Rh negative, it means they don't have it, so you give them a little negative. So let's say you have the A protein plus the Rh protein. That means you are A positive. If you have the A protein but no Rh factor, then you are A negative. Let's see. And I'm not going to spend time talking about this. There are differences between your body's reaction to the Rh antigens and the ABO antigens, but it's not going to be on the quiz or the exam, so we'll just skip right over it. And then again, transfusion reactions. This just goes into more detail about what happens if you get an agglutination reaction in your bloodstream. So then we get into blood type testing. This is where things get weird. What you need to remember is that now we are adding antibodies to the bloodstream that we bought from a company. So in this scenario, we're not talking about when we're looking at anti-A, anti-B, anti-RH, right? These aren't the antibodies that are supposed to be floating around in your bloodstream. These are the antibodies that we got out of a bottle that we're adding to your bloodstream because we're trying to figure out which antigens are there. So you're going to see the way the test results read is opposite to how you would think the antibodies would work in reality in somebody's actual bloodstream. Not reality, it's all reality, but the test is a different highly different scenario, an artificial construct, as opposed to what's supposed to be in your bloodstream. So the way it works is that you take your blood. In this case, they're just doing AB. I added the RH over here myself. So you have 2 drops of blood that initially started out looking like that, a solid drop of blood. You added anti-B agglutinin to this blood, anti-A agglutinin to that blood, and you get clumping. So it should look like this here. This means that the anti-A antibody that you added to this person's bloodstream stuck to the A agglutinogen. That means this blood has the A agglutinogen, but here nothing happened, so it doesn't have the B agglutinogen. So this would be type B blood. So now, because we're adding antibodies that we bought from a company that aren't supposed to be in your bloodstream, when you see the reaction, the antibody matches the blood type. So here, type B, again, we took blood from this person, put a drop here, put a drop there, added the anti-A antibody. There was no A for it to stick to, so nothing happened. Here we added the anti-B antibody, so it clumped, which means the anti-B antibody found the B agglutinogen, stuck to it, caused the blood to clump. This is type B blood, right? Clumping in response to both antibodies means both of the agglutinogens were present in the bloodstream, so you get clumping in both. So this is AB blood, no clumping is O blood. And then if you want to know whether you're A positive or A negative, you have a 3rd drop of blood, like in this picture here, and you add the anti-RH antibody to that. So if the blood stays looking like regular blood, that means the anti-RH antibody or agglutinin did not find an RH agglutinogen to stick to, so this is RH negative. So if I took this and put it there, we would have type A negative blood. Here, right, we add, the anti-RH antibody to this blood, it clumps. That means that the protein is there. So if I stick this, let's say here, right, now we've got B positive, AB positive, B negative, AB negative. I put that back there. So that's how it works. So for the quiz, you want to know what is an agglutinogen, and then based on blood type, you want to know which agglutinins are going to be in that person's blood and which agglutinogens are going to be in that person's blood. Don't worry about the transfusions. And then also know if somebody's AB positive or AB negative, what does that say about the presence of the RH protein? Then, if I present you with some results, something that looks like this, right, without the name there, you need to be able to look at the results and say this is B negative blood or O negative blood. And that, if you watch the intro video already, that's how you interpret the blood test results that are in the intro video. That's it. I hope it makes sense. And yeah, that's it.